Today we'll finish two topics. The first one is a shorter one, the diagnosis and treatment of virus infection. So we had a similar topic before when we talked about bacterial infection, diagnosis and treatment of bacterial infection. Well, this one is for virus. It's a similar structure, but the content is different. Sorry. Okay, for viral infection diagnosis, and yeah, of course, the first thing is that we should analyze the specimen. It's really hard to diagnose your patient only based on the symptoms. And you want to collect the specimen and then analyze it. No matter what material you are working on, like the antigen or antibody or nucleic acid, but you won't have the material. That's the specimen. We should select the proper specimen depends on the specific disease symptoms. That relies on your professional experience, and you decide the proper specimen to collect. And also, we want to collect the specimens as early as possible following the onset of the disease. So this is also important. Once you have your patient in your clinic, you want to collect the specimen as soon as possible. And also, the specimens for viral isolation should be placed on ice. This is critical. Anything you want to share? Oh, nice. We should place the specimens on ice because, you know, when you collect the specimen, it won't be in an environment which is completely sterile. And it's possible that the specimen you collected is contaminated by bacteria, especially the normal flora from the patients. And placing them on ice can inhibit the bacterial growth. And after that, we should transport the specimen to laboratory for further analysis. And we should do this as soon as possible. And also, usually for the specimen for virus analysis, we want to add some antibiotic to it. So antibiotics are usually used in the specimen for virus detection. So yeah, you understand what's the function of antibiotics? You? I don't know. It's not on the TV. So antibiotics is, we use them to kill bacteria. So we use antibiotics to kill or inhibit bacterial contamination in the specimen. So this is the key point for collection of specimens. And once in the lab, you have the specimen, you can examine the specimen, and similar like the specimen for bacterial infection, you want to have a quick check. Quick check, like you stain the specimen, you observe it with a light microscope for bacteria. But for virus infection, well, simply staining and observe with a light microscope does not help. So we rely on electron microscope to see the morphology of viral particles. But usually, it takes a long time to prepare the electron microscope sample. You need to fix it, stain it, and then apply it to the electron microscope. and usually it takes several days, even a week, a couple of weeks to get the results. So it's not easy. But you can also use light microscope, the fluorescence microscope. Use the immunostaining. You stain your specimen with specific antibody, which is labeled with fluorescence. And so that you can check the fluorescence with a fluorescent microscope. And this result, you can get it pretty quick, within several hours or a couple of days. So you can use this immunofluorescence staining, and this is also specific immunoassay. And sometimes you want to culture the virus. This is not widely used in hospital because you don't want to culture the virus for every patient. It's very expensive and probably not useful. But if there is a pandemic, if there is an outbreak of certain virus, you want to determine the type of virus, and you need to purify the virus and culture it. And we can culture viruses in different ways, such as we can use the cell culture. We culture the host cell in this flask, and then infect the flask with the virus in the specimen, and so that you can collect more particles. You can also culture the virus with a chicken embryo. Chicken embryo incubation allow you to harvest large number of viral particles. Like the vaccine we are using against influenza, most of the vaccines are cultured in chicken embryo. It's low cost and can produce large numbers. And sometimes you can also use animal model to infect some animal. And this is limited for certain viruses which cannot be cultured in the cell or in the chicken embryo. And you need to use special animal, especially the primate animal, to amplify the virus, such as the HBV and HCV. We'll talk about later. So this is the virus culture. Okay, to detect the viral antigen, we use immunoassay. We use a specific antibody to recognize the antigen. And this can be carried out in different ways, such as the one we mentioned, the immunofluorescence, and also you can also use the enzyme immunoassay. Either the ELISA, we talked about before, we did that before, or the immunohistochemistry assay. And both of them are based on the enzyme immunoassay. And yeah, previously, people use a lot of radioimmunoassay to label the immunoreaction with radioactive material. But nowadays, we don't use such methods a lot. At least as I know, in China, it's not popular anymore because using radioactive material is kind of dangerous sometimes. So this is antigen detection. And you can detect the presence of nucleic acid, either DNA or RNA. You can run PCR to examine it. If it's DNA virus, you can run PCR directly. If it's RNA virus, you need to run the reverse transcription step before you run PCR. You make complementary cDNA, then amplify the cDNA. And you can also use nucleic acid probes to detect the presence of certain sequence or the nucleic acid hybridization. And all these methods are being used in the hospital. Okay. And you can also get the patient's blood and serum and, and detect the presence of certain antibody in the patient's serum. So with the antibody, we can do like neutralization test. Neutralization means that the antibody in the patient's blood can inhibit the infectivity of a virus by blocking its contact to the host cell receptor. So neutralizing antibody binds to the viral particle and can block the infection. We can use this test to detect the presence of neutralizing antibody. And we can also run the hemagglutination test. Hemagglutination is this way. Some viruses, they carry the hemoglobin. You mix it with a virus, like the influenza virus, the HA protein on the surface can bind to the red blood cell. And the viral particle can link a lot of red blood cells together. This will cause the hemagglutination to link a lot of cells and viral particles together to form a big complex. The big complex in the well will precipitate out as the whole body together. So you will see a membrane covering the bottom of the well. This is hemagglutination. While the inhibition test, you mix the virus, red blood cells, and meanwhile, you add in the patient's serum, which contains the antibody. The antibody will bind to the viral particle so that hemagglutination cannot happen anymore. So this is the hemagglutination inhibition test. Inhibition is due to the presence of neutralizing antibody, which inhibit the hemagglutination. And this method can also help you to detect the presence of antibody in the patient's serum. And we can also run the complement fixation test. I showed you the mechanism of this method before. Complement fixation assay. Okay, this slide gives us a quick summary on the methods of diagnosis of viral infection. Okay. Anyone, any girl. Okay. This get the specimen from the patient, and there are several different methods like culture. This is the PCR, and here the electron microscope, and this is the in fact the cells, and you can see the morphology of the cell change, and also this fluorescence microscope, and also immunoassay, such as the Western blot. It's another way, depends on the antigen antibody interaction. Okay. Okay, next we'll talk a little bit about the antiviral therapy. Unlike bacterial infection, we can use a lot of antibiotics to kill or inhibit the bacterial growth. Well, for viruses, okay, all the antibiotics cannot work. This is this consistent with your life experience? Like when you have a common cold, will you take antibiotics? No, no. Anyone else? If you have a common cold, you need to take antibiotics. No? Yeah, the answer is no, right? No. But a lot of doctors, especially doctors here, who are not professional, if they have a patient coming into the clinic or to the hospital, especially the little kids, they give them antibiotics. And they suggest that, well, during the common cold, it's possible that there is a recombination complication of bacterial infection. So they give them antibiotic to prevent bacterial infection, which is not logic, as you use antibiotic before any possible bacterial infection, which is not helpful. But that's common here in China. In further years, when you go to the hospital, if you see such phenomena, well, keep that in mind. It's not reasonable. But for viral infection, we do have some medicine, some mechanism to inhibit the viral replication, such as the antiviral chemotherapy, and we can also use some cytokines, such as the interferons, and we do have the viral vaccines available, some of them available, so you can use. The first one is the chemotherapy. Chemotherapy, according to the name, okay, we use some chemical, okay, some chemical to inhibit the viral replication, such as we can use the nucleoside or nucleotide analogs, the reverse transcriptase inhibitor, this is for HIV, some protease inhibitor. For a lot of viruses, they should use some protease to cut the big polypeptide into a lot of proteins, such as the COVID-19. There is a protease inhibitor, and also other types of antiviral agents. The first one is the nucleoside and nucleotide analog. And again, this is for HIV. And we know that the first step after uncoating, release of the HIV RNA into the cytoplasm, the first step is the reverse transcription to make DNA strand. Okay, this is DNA strand. And we know this is a simple structure of DNA. You have a ribose, the sugar, and that's the base, right? The base to form the hydrogen bond between the two strands. And the backbone contains the phosphate group. So ribose phosphate, ribose phosphate, ribose phosphate. And the extension of the DNA is always on the 3'. Remember that? Always on the 3', because on the 3' on the ribose, the number 3 carbon, there is a hydrogen bond. Sorry, this group, hydroxide group, right? Hydroxide group, yeah. This group can react with the phosphate group, okay, to link it to this phosphate group. Okay, this is naturally happening in our cell, but the analog we use is this molecule. At the 3' end, it's not a hydroxy group, it's N3 group, okay? Once this analog is, okay, once this analog is integrated into the DNA chain, the DNA cannot extend, so you block the DNA synthesis, okay? No more extension. So we use this analog to inhibit the reverse transcription. Okay. Another type of inhibitor is a reverse transcriptase inhibitor. It's also at this step, we inhibit the activity of the reverse transcriptase. So no more DNA made. So you can design different type of inhibitors, such as this one. This inhibitor binds to the DNA strand, the DNA molecule, okay, which can block the reverse transcription. While this inhibitor actually it binds to the enzyme directly, binds to the reverse transcriptase directly, so that no DNA can be made. And we can design those chemicals, those inhibitors, to block the reverse transcriptase activity so that we can inhibit the replication of HIV virus. Another type of inhibitor is the protease inhibitor. When we introduce the genome of viral pathogens, we know that in the viral genome, it's a common phenomenon that sometimes in the viral genome, it links a lot of genes together. So links several genes together. And when the genome is transcribed, it will be transcribed in a single molecule, then translated in a single molecule. After translation, there will be a big, long polypeptide. But this big, long polypeptide is not functioning because it contains a lot of proteins in it, okay? A lot of proteins together, linking together to form a big polypeptide. Okay, to let those proteins function, you need to cut the polypeptide into small pieces. Each piece is a functional protein. Okay, again, this is for HIV. The GAG protein contains, okay, after cleavage, this okay, let's see the name. P17, 24, P7, P6, 4 different proteins. And this relies on the activity of this protease. And this protease is also encoded in the HIV genome. So we can design an inhibitor, protease inhibitor, to bind to the protease, okay, inhibit the protease activity. Once the protease is inhibited, the polyprotein cannot be separated to function so that we can inhibit the viral replication. It's similar in COVID-19 pathogen. It relies on the protease activity, while in the medicine, which is available now, it contains this, not this, it contains a protease inhibitor, which can inhibit the COVID-19 virus replication. And we do have it, which company? The American company. Anyone know the name of? Anyone know the name of that company? Pfizer. Yeah. Pfizer have medicine. We call it P medicine, and it contains the protease inhibitor. Okay. Okay. Another type of medicine we can use is the interferons, or it's a type of cytokine. Okay. And when we learn immunology, we know that there are 3 major types of interferons: interferon alpha, beta, and gamma. And I think you learn interferon gamma a lot, right? For your immunology. Interferon gamma is produced by lymphocytes, like T cells or NK cells. And it belongs to the type 2 interferon. The major function of interferon gamma is for immunoregulation. Okay, it regulates the activity of other immune cells. So that's the interferon gamma, like activate other immune cells. But there is another type called type 1 interferon. But in this type, interferon alpha and beta, and they can be produced by many different types of cells, many cell types, not lymphocytes, but a lot of cells can produce interferon alpha and beta. And the function of these type 1 interferons are the antivirus activity. Here is how it works. Interferon molecules do not inhibit viral replication directly. Okay, it functions in an indirect way. This is a host cell, okay, and this is a virus particle. It infects this cell, okay, and this cell is infected for sure. But during infection, this infected cell will turn on the expression of interferon molecule, interferon alpha and beta, okay? The transcription, then translation, then secrete interferon molecules to the extracellular space. Then the interferon protein, the cytokine, would bind to its receptor to influence some neighboring cell, okay, neighboring host cell. In this host cell, it receives the signal from this interferon molecule. Then it activates the expression of antiviral protein. Okay, a lot of antiviral proteins. And those proteins would, well, if there is virus infection, can degrade the viral messenger RNA and also inhibit the protein synthesis. Once the protein synthesis and messenger RNA is inhibited, the virus cannot replicate in this host cell. But this is how the interferons function, inhibiting viral replication. Okay, it's in an indirect way, not directly inhibiting the viral life cycle. Okay, this is the interferon molecules. Okay, this is a quick summary of the possible antiviral therapies. On the left, okay, on the left, let's see, only for HIV virus. And you can see that along the life cycle of the HIV infection, okay, attachment, penetration, uncoating, biosynthesis, and then releasing. At each different step, you can design the inhibitor, okay? Inhibitor. Here, inhibitor. So right now, over 40 years, we do have a lot of medicines available to inhibit HIV replication on different targets. Okay, at each step, you can pick some target to design the medicine. So right now, to cure, not cure, to help the HIV infected persons, we can sometimes combine several medicines together, and we use it as a cocktail. Because in a cocktail, it can inhibit the viral replication better, and also we can avoid the resistance to happen. Okay, if one medicine cannot work, other medicines can still inhibit the viral replication. So this is for HIV. Well, on the right, it's a general method for other antiviral therapies. Like here, you can use interferon alpha and beta, and some medicine like influenza A. Okay, here it inhibits the uncoating. And in China, there is a medicine which is widely used. It's called the oseltamivir. In Chinese name is oseltamivir. This is the enzyme. This is the inhibitor which can inhibit the activity of neuraminidase. Neuraminidase is the enzyme which is required to cleave the molecule to release the viral particle from the host cell. Okay, but the inhibitor can inhibit the enzyme activity so that the virion particle cannot be released from the host, so that you can inhibit the influenza virus. I'm glad that no one is infected by flu this year, because right now in the hospital there are a lot of patients suffering from flu infection. Okay. So this is the topic on diagnosis and treatment. Some of you guys ask me where you can get the vaccine, no matter what, like flu vaccine, HPV vaccine, and also what else? There are some vaccine you can have. We have some medical service center here, close to our campus, and you can check it on the map. And they have the vaccine available, especially the flu vaccine. And last time I went there, I asked them, for you guys, if you want to receive some flu vaccine, you can get it, but you have to pay. It's not free. It's like 110, 150 RMB. It's not too expensive, but make sure that you bring your passport with you, because you don't have the national ID in China, but your passport will work. Okay. It's not far from here. Okay. So this is for diagnosis and treatment.